Hope everybody had a great break. We're back here through the last couple weeks and we're almost done. Okay. So today we're going to move into the heart, but before we do that, I want to do a brief overview of smooth muscle. So far, done everything we possibly know about skeletal muscle, hopefully. Smooth muscle is our first type of involuntary muscle, so muscle that's not under our conscious control. And cardiac muscle, where we'll spend most of our time talking about the heart. This is a brief primer on that muscle type that we're going to be skipping over it a little bit until we get to blood vessels. Look at the difference between our skeletal muscle and our smooth muscle. So when we were talking about our muscle cells, muscle fibers, we saw one of the things that we noticed about skeletal muscle was they had a lot of different nuclei because they merged together into those fibers. And they're also striated, so striped, because they have those sarcomeres in them that create those characteristic banding patterns based on where the actin and myosin do and don't overlap. So when we're thinking about smooth muscle, it's going to look quite different if you look at it under a microscope. So you'll see that we have a more kind of typical structure to the cell. They're these kind of teardrop shapes. We don't have this banding pattern, so we don't have striations. And this is going to make a difference for how they function and how we control them. So smooth muscle is found in internal organs and blood vessels. So think like arteries, like your GI tract, places like that. So it's under involuntary control. It means it's controlled by the autonomic nervous system. So sympathetics and parasympathetics we should be thinking here. Muscles are these spindle shapes or teardrop shapes. Smooth muscle is small. It's about a tenth the size of skeletal muscle. So like skeletal muscle, it's still going to have actin and myosin, but they're not structured, bless you, in sarcomeres anymore. So instead, we're going to have these fibers attaching to these structures that we're going to call dense bodies. So dense bodies are kind of these nodes between this kind of web-like structure of actin and myosin and smooth muscle. So we have thick filament and thin filaments overlapping and coming together at these kind of angles, which means that when smooth muscle contracts, the shape of it changes in a subtly different way than when we see our skeletal muscle contracting. And a lot of the time when we see smooth muscle, it's going to be surrounding a cavity. So that'll also be a difference that we'll think about when we think about it like narrowing a blood vessel or kind of sphincter-shaped muscles. So we still have a sliding filament mechanism of contraction with just subtle differences. Actin and myosin are longer in smooth muscle. The myosin heads don't have a bare zone, so they're just the whole length. We have a longer range of contraction, and the contractions happen more slowly. So it's involuntary, kind of slower process. So involuntary, so it's the autonomic nervous system, sympathetics, parasympathetics, and like everywhere else have excitatory, or I should say, like in our nerves, we can have excitatory and inhibitory effects, whereas in skeletal muscle, we were always kind of exciting. We were always creating a contraction when we sent an impulse. So we're going to have differences in the response depending on the receptor types. So when we see smooth muscle, again, we'll be dialing down on those receptors. Remember, like our adrenergic receptors, things like that. So we are going to have two types of smooth muscle. So our neurotransmitters are going to be released from varicosities. And we saw those are those like spaghetti fingers kind of spreading across that we saw when we looked at the structure of our autonomic nerves. So we're going to see this diffuse binding to the receptors. Our two categories are going to be single unit smooth muscle and multi-unit smooth muscle. And the difference here is going to be kind of the relationship of the nerves to the muscle cells themselves. So single unit smooth muscle, we're calling it single unit because the whole muscle, like all the muscle cells, contract as a single unit, which means that we're just going to have this one neuron with its various swellings, those varicosities, over the surface of the smooth muscle. So we're going to have those neurotransmitters spreading to the cells that they're contacting, and then we see a lot of gap junctions. So that's how the neurotransmitters and signals are going to spread throughout the smooth unit, or the single smooth unit muscle. This is going to happen in the intestinal tract, it's going to happen in blood vessels, the respiratory tract. So these muscle fibers are activated synchronously, so together, they're moving together as one unit. They don't each have like their own nerve. So those are pictures of those gap junctions, which is just that connection cytoplasm to cytoplasm, like a hole straight through. A cool thing about smooth muscle is that sometimes we have these cells we call pacemaker cells, which we're also going to see in cardiac muscle in our heart unit. So what a pacemaker cell is it undergoes spontaneous depolarization. So when we say spontaneous depolarization, we really mean that it doesn't need an impulse from a nerve in order to create this change in its voltage so that it can contract. That's going to happen kind of in a wave. So spontaneously, they have this internal pace. It's just the quality of how that cell works. So smooth muscle does sometimes have these pacemaker cells. So the rest of this is mainly just describing the structure. So we can see this one neuron lying over top. So the direct innervation is only going to a few cells. We have the contraction happening because of those gap junctions connecting everything. Another feature we'll see about smooth muscle is that we have this thing called tone. So when we say a muscle has tone, here we mean that it has a certain level of contraction. It's not at its most relaxed, longest state at rest. It's slightly contracted. So we see increases and decreases in that tension from that kind of middle level of tone. Smooth muscle also has what we call a stretch reflex, which is where you have this sudden and prolonged stretch inducing relaxation. So it's kind of like a bouncing back kind of thing that we have happening there. The other thing that we have here is that instead of having a contraction or not having a contraction, like all or nothing, a smooth muscle can have graded contractions. So like slight contraction, slightly stronger contraction, and so on. And this isn't happening because of motor units and recruitment. This is happening because of different levels of calcium. Multi-unit smooth muscle, we just see those autonomic nerves diving between our smooth muscle cells, which is why it's called multi-unit, because each fiber then has its own innervation. And so it could contract on its own without the neighboring cells. So we're not going to see really any gap junctions. We might see these contracting independently of each other. And it's also true that these fibers don't happen to have tone, and they do kind of have their own type of recruitment because we're contracting these cells, and then this cell, then those cells. So that leads us into the heart.
We're not really in control of skeletal muscle.
What did we miss? Sorry.
From Friday, we
I thought that was where we left off. Did we miss this stuff? Oh, sorry. Okay. Can you tell me where we left off? I thought we talked about the different types of fibers. Did we not talk about this exercise?
Control the skeletal muscle.
Okay, maybe we are skipping it then. Sorry, unless you see me clicking through it, but
Nope.
Yeah. Okay, yeah, I'm gonna skip it so that we can move into the heart. All right. So we're gonna start out with heart anatomy, basically, which you will get into in lab in A&P too. But to understand heart function, we can't really talk about it unless you know the pieces of the heart. So we're going to be thinking about the heart itself and its different compartments, basically. We're going to be thinking about the blood vessels leading into it and coming out of it. We're going to be thinking about the blood moving within it as well, because we're going to see that the function of the heart is really dependent on pressures in different parts of the heart. And when I say pressures, the thing we want to immediately think about now, pressures are going to work just like our chemical gradients work. We're going to be moving from areas of high pressure to areas of low pressure. So that's what we're thinking about with the blood. So the overall purpose of our cardiovascular system is to transport different substances around the body. So a lot of that is going to be oxygen. We know that oxygen is really important for most of our cells to produce ATP. We're also thinking about nutrients, for example, glucose. We're going to be removing waste cells as well. So specifically in the liver and kidneys, we're going to be thinking a lot about waste products. And we're also moving our hormones around, right? We remember that the endocrine system, referring to the fact that we're sending hormones through the blood to distant areas. So they have to travel through the cardiovascular system. We also have immune cells going around. We have different clotting proteins allowing us to heal injuries to the cardiovascular system. We will talk about clotting in this class. Right. So starting out with the heart, actually, like I linked something here. Okay, so we're going to watch blood move through the heart, and then we'll take a look at the picture and key in on these different places.
Oxygen poor blood, blue blood, returns to the heart after circulating through your body. The right side of the heart, composed of the right atrium and ventricle, collects and pumps the blood to the lungs through the pulmonary arteries. The lungs refresh the blood with a new supply of oxygen, making it turn red. Oxygen-rich blood, red blood, then enters the left side of the heart, composed of the left atrium and ventricle, and is pumped through the aorta to the body to supply tissues with oxygen. 4 valves within your heart keep your blood moving the right way. The tricuspid, mitral, pulmonary, and aortic valves work like gates on a fence. They open only one way and only when pushed on. Each valve opens and closes once per heartbeat, or about once every second. A beating heart contracts and relaxes. Contraction is called systole, and relaxing is called diastole. During systole, your ventricles contract, forcing blood into the vessels going to your lungs and body, much like ketchup being forced out of a squeeze bottle. The right ventricle contracts a little bit before the left ventricle does. Your ventricles then relax during diastole and are filled with blood coming from the upper chambers, the left and right atria. Then the cycle starts over again. Your heart is nourished by blood too. Blood vessels called coronary arteries extend over the surface of your heart and branch into smaller capillaries. Here you can see just a network of blood vessels that feed your heart with oxygen-rich blood. Your heart also has electrical wiring to beating. The electrical impulses begin high in the right atrium and travel through specialized pathways to the ventricles, delivering the signal to pump. The conduction system keeps your heart beating in a coordinated and normal rhythm, which in turn keeps blood circulating. The continuous exchange of oxygen-rich blood with oxygen-poor blood is what keeps you alive.
So I find seeing those movements helpful as we talk about the parts of the heart, because otherwise it's just memorizing a bunch of names, which is not always the most fun. Okay, so let's go ahead and trace that path of blood through the heart. Okay. So what we're looking at, we're looking at a cut through the heart, so it's transected. And here we have our right side, and here we have our left side. Okay. When we think about the heart, we can kind of think about it in quadrants, although it doesn't look like that in the picture. So we could just draw a circle and split it twice, or if we want it to really look heart-shaped, we can draw a heart, and then our walls are good like that, right? Okay. So starting with the right atrium, so that would be here in this picture, here. This is where we have blood coming in from the body that has had all the oxygen used up, right? So our deoxygenated blood coming in from our veins into this right atrium and pulling there. So here we can see those veins. So our superior and inferior vena cava have blood pooling into the right atrium. I remove that second arrow just so we can have one kind of smooth directional arrow, but it is coming from both the upper body and lower body. After the right atrium, the blood, as it fills there and pressure builds, it's going to start to move into the right ventricle. We're coming in and we're going down first, basically. So atrium to ventricle. So this is our right ventricle here. And to pass between the two, we're going through what we refer to as our right AV valve, or the tricuspid valve. So the cusps, here we can see like two of them. There would be a third if we were in 3D. So this is the cusp. That's another cusp, and they're connected to these muscles that we're going to see are called papillary muscles coming out of the walls of the ventricle. So we've moved from the right atrium to the right ventricle. All this blood is still deoxygenated as we've moved through the right side of the heart. We don't get oxygen until we go to the lungs, which is what we're going to do when we contract the right ventricle, right? So systole, contraction. In systole, we contract our ventricles. Blood is going to move out of the heart. The blood from the right ventricle is moving out through the pulmonary trunk and the pulmonary arteries. And this is the only time you're going to see arteries colored blue in the body. So an artery technically means something going away from the heart, not actually talking about the oxygenation state of the blood. In this case, this is the one case where we have blood going away from the heart that doesn't have oxygen in it. Okay? So try not to get confused about the fact that these are arteries colored blue. Okay, so we have the blood coming out, and I'm gonna do it over here just so we don't cover over our left side of the heart yet, right? So we're going to along. One long. Okay, so that's our pulmonary trunk and our pulmonary arteries. Now, as we circulate around basically the walls of the lungs, we'll talk about exactly how blood gets oxygenated in a respiratory unit. But we're soaking up oxygen. Oxygen is binding into hemoglobin, which is why we have that color change in the blood. It may not exactly be blue when we're looking at venous blood, but it is darker. We're turning really bright red when we get it to the arteries as we bind in that oxygen. Okay. So then our blood is going to have to come back to the heart. So this is where we're going to see red veins coming into the heart. Right? So this is the one time we're going to have really highly oxygenated blood in a vein. We're actually coming all the way here into the left atrium. So I'm going to draw this in kind of a loop like that. The left atrium. So now we have oxygenated blood in the left side of the heart. As it builds up, it's going to pass down into our left ventricle. Okay. And as it does this, it passes through another valve. So we're passing through another atrioventricular valve, or AV valve. In this case, this one is called the mitral valve, the bicuspid valve. And then during systole, when our ventricles are squeezing, the left ventricle then squeezes this blood out the aorta. So that big arching structure is our aorta. So you have it moving like that and out to the rest of the body. So that's the basic sort of flow of blood we're going to expect to see. The right side of the heart is going to be full of deoxygenated blood. The left side of the heart is going to be full of freshly oxygenated blood. And these great vessels, these arteries and veins coming out, are carrying blood either to the lungs or sending it out to the rest of the body. So that's the kind of basic structure of the heart. Otherwise, atria are on top. So you have a right and a left atrium. Ventricles are on the bottom. So you have a right and a left ventricle. But the flow of blood is down one side and then down the other side. So right to right, left to left, atrium to ventricle. Oh, yep.
So since we're focusing more on the physiology side of things in this class,
Mhm.
in this section, we don't need to know like all of the names, right? Is it just like we're gonna focus more on like the AV valve rather than?
It's not like I'm going to ask you to label a picture, but we are going to talk about like, so pressure that builds in the right atrium during this phase and then has to pass to where or something like that. So we are going to be following the blood. Yeah, that's why I was trying to show you with the arrows. But so we want to focus on like the flow, right? So we'll be focusing on what way the blood flows through which structures, and then also we'll be talking about the electrophysiology, so the electrical system that causes these contractions. Yeah. But basically, yeah, we're not learning the names for themselves, but to talk about what's happening at any time during this contraction, we need to know at least some of those names. All right. So when we're talking about blood vessels in the cardiovascular system, we have different levels as well. And as we move through, we'll talk about different parts of the physiology and different types of blood vessels. So we're starting here with the heart, but after the heart, we're going to pass into the arteries. So the arteries are large, they're branching vessels, they're coming off the heart, and then we'll have branches of the aorta as well, count as arteries. So those are really what you're seeing in the lab when you see parts of the cardiovascular system. You're either looking at an artery or a vein. But we have more branching that we don't tend to see with our naked eye. So after the arteries, as we get smaller, we start to have arterioles. So our arterioles are smaller branches, but they're specific characteristics. So they're going to have high resistance that we can vary with smooth muscle. So when we say resistance, it's kind of like friction. There's like a bunch of drag on the blood. And we're going to see that in the arterioles, that's really where we're going to be controlling kind of the blood pressure, but really how much blood is getting into different types of tissues. Our capillaries are going to be the site of exchange between blood and tissues. So we'll see, for example, oxygen and carbon dioxide passing through capillary walls. And then we'll start returning blood. So on the other side of a capillary bed, we'll feed into a venule and then into a vein, and that would be our deoxygenated blood. But this is a closed system, so we're not going to have blood leaking anywhere. It just keeps circulating through unless there's something wrong. So blood has to go through all these pieces. So have you guys seen this picture already in Phys Lab? Okay, so when we're thinking about composition of the blood, we have different components here. We have our red blood cells, or erythrocytes, and we'll eventually talk about their life cycle. They're the ones transporting our oxygen and carbon dioxide. We have our leukocytes, the white blood cells. So as you know from your histology lesson in phys lab, we have a lot of different types, but their general role is to defend against pathogens. We have platelets, which are tiny little cell fragments that are going to be important in clotting. We have our plasma, which is extracellular fluid, and various solutes. So for the cardiovascular system, as we're concerned about the heart, we're going to be thinking about this as two circuits. So we're going to think about the right side of the heart as part of the pulmonary circuit. So pulmonary is your lungs, right? So the pulmonary circuit is circulating the blood to your lungs to get oxygenated and then back in order to feed the left side of the heart. And our systemic circuit is going to be controlled by the left side of the heart system, like the rest of your body. The systemic circuit is pumping blood out to all your various tissues and organs. Now, we refer to the type of flow in these different circuits using like physics terms, I guess this is. So we refer to flow through the cardiovascular system, any like one of these as series flow. Sorry, so we have to flow through the pulmonary circuit and then the systemic circuit. So that's the series. We have a 1 and 2, they're in order. But once we're in a circuit, really within the systemic circuit, this is where it makes more sense. We're going to have parallel flow, meaning that blood is going to be flowing like to your liver at the same time as it's flowing to your GI system. It doesn't have to flow through one organ and then another organ and then another organ. They're all kind of getting their independent blood supply at the same time. So series flow has to happen from the pulmonary circuit and then through the systemic circuit because the cardiovascular system is a closed system. So it has to go through these things in an order. Otherwise, it wouldn't get back to its starting point. We'd have it pooling somewhere, or we wouldn't end up with it getting oxygenated before it goes through the systemic circuit. So series means we do 1, then 2. We have to do pulmonary, then systemic. So parallel, what we mean is that it would look something like this if you were to diagram it out, right? Like we have parallel lines, right? They're all branching off on their own from the arterial system. Okay. And this is important because it's going to allow us to regulate blood flow to each of these organ systems independently, which, as we've kind of seen as we were talking about skeletal muscle and exercise, is important because you don't always want an organ to have the same amount of blood or the same like fraction of the blood. Sometimes it's going to be more important to send blood to your gut. Sometimes it's going to be more important to send blood to your skeletal system. And we want to be able to control that flow. We're going to be able to do this because we can control blood flow really well using the resistance of the arterioles, controlling them. So we have blood flow to any given organ. Let's pick these ones. Let's pick skeletal muscle here. So we'd be going through an artery, then we're passing through arterioles into the capillaries. These kind of spindle structures are our capillaries, as we can see that we're slowly turning from red to blue as we allow oxygen to leach into the tissues, then passing into our venules and into our veins. So that would be happening at the same time, right? The kidneys would be getting blood at the same time as the skeletal muscle gets blood, the same time as the skin gets blood. We don't have to go from the brain to the liver to the kidney and so on. You can see that there are a couple exceptions. I probably shouldn't have picked liver and GI tract because sometimes we do have blood supply connected between the two. When you look at the abdomen in lab, you'll see why that is the case. There's a special like portal system, kind of like we saw in the hypothalamus and anterior pituitary. But in general, we want to think about these things as happening in parallel. So go ahead and make your own little diagram of what a systemic circuit to the organs would look like. And write down for yourself why that has to happen. Maybe start with the heart, right? Because we're interested in the heart mainly. Heart. So in our systemic circuit, how's the blood flowing? Series or parallel? Parallel, right? So in parallel, meaning we're going separately to, for example, a skeletal muscle, skin cell, no way for you to know that's what I drew, but that's fine, or our GI system in the liver. So they're all separately returning their blood. Here's a venous system, so a venule and then a vein. And we contrast this with the overall cardiovascular system, in which case we would add on our pulmonary circuit, right? It should look like this, going to the lungs, and then coming back. So here we draw some nice lungs. All right, so the fact that we go through pulmonary circuit and then systemic circuit, that's our series flow. Does that make sense, the difference between series and parallel? If you've ever built circuits in a lab, that's where we're drawing that terminology from. But it's really just, do they go in order or do they go all at once?
So the heart as a whole is that parallel blood flow, since different sides of the heart are doing different things at the same time, or is that?
The inside of the heart we would think of as series because it goes through right and then left. But here we have parallel flow to the heart when we're talking about feeding the muscle. So the heart actually, what we're talking about most of the time is the blood inside the heart, but that's not where the muscle is getting its oxygen. We also treat the heart like as its own organ system in terms of blood flow. So the heart supplies itself as well using these coronary arteries. So in that sense, it would be in parallel when we're talking about feeding the muscle of the heart. But if we're thinking about it as its like role in the whole cardiovascular system and the flow of blood, that would be more kind of series role. Perfect timing for the question, because here we see those coronary arteries. So this is just a note here that we do supply the skeletal muscle, or sorry, the cardiac muscle from the outside using these arteries. So if you, or anyone who's had like a coronary bypass, we're talking about these arteries that are feeding the muscle of the heart, not the internal blood flow inside the heart. So you'll learn all their names in lab, so we're not going to worry about those right now. We just want to know in general that that's how that muscle is getting blood. These would have their own capillaries, and they actually have their own venous system as well. This is actually kind of the most important to your body thing, I mean the closest. So they branch directly off the base of the aorta here. So that's where that blood flow is coming from. So overall, heart location is in the thoracic cavity, has a diaphragm below it, which is going to be helping us control breathing and the respiratory system. Usually about the size of a fist, weighs 300-ish grams. What we're thinking about now is the sort of layers of the heart. So kind of like we thought about layers of the muscle and things surrounding it. The heart is in a sac, a membranous sac that we call the pericardium. And the point of that is that it allows the heart to expand and contract, right? So to relax and contract distally and diastole, such that it doesn't have much friction from the outside, right? It's not good if you can't move your heart easily in order to allow for this blood flow. So pericardium is lubricating the heart and allowing for it to move freely. I'll also see that this is a potential place where we could have pressure build up in this sac pathologically, which would put pressure on the heart, which is problematic. Within the sac, we have the heart itself. So the outside layer of the heart, we call the epicardium. It's basically a membrane that's gliding across the pericardium. So it's the external membrane. And then inside, we have the muscle itself. So that's what these are, right? This is showing us the myocardium inside the walls. Then we have a layer of endothelial cells. So kind of like epithelial cells, but they're on the inside of a cavity, lining those atria and ventricles. So epi, next to outside, myo, the actual muscular part, and endo, meaning inside. So that's that interior layer of cells. Okay. Which gets us to why the blood flow is happening. So the fact that we have this muscle contracting, diastole, the relaxation, and systole, that contraction, means that really what we're doing is controlling the pressure in the chambers of the heart. So in terms of blood flow, our blood is always going to move from an area of high pressure to low pressure if it can get there. That's the important piece with the valves. So the valves are going to be blocking us basically from backflow, because we're going to see that as blood moves from the atria to the ventricles, that raises pressure in the ventricles. We don't want blood flowing back from a ventricle to an atria. So that's what's going to be important about those valves. But always, if it can happen, if there's an opening, we move from high pressure to low pressure. So the normal direction of that flow is from the atrium to the ventricle, from a ventricle to an artery. Okay. So right atrium to right ventricle to pulmonary arteries, left atrium to left ventricle to the aorta, which is an artery. The valves preventing us from going from the ventricle to the atrium, we actually have some valves in the arteries as well that prevent blood from flowing from an artery into the ventricle. None of these valves are opening or closing based on muscles. They're all opening and closing based on pressure gradients, but we will see some muscles associated with the AV valves. And all those muscles are doing is like preventing the AV valve from like prolapsing, from blasting backwards from the sheer degree of that high pressure. Taking a look at the AV valves first. So we have our tricuspid valve, which is on the right. So here we can see the three, maybe we can see three cusps. 1, 2, 3. Okay. 3 cusps on the right, 2 cusps on the left. 2. Okay. So what's happening is that when we have high pressure in the atrium, as pressure rises in the atrium, There isn't very much blood yet in the ventricle, so it's an area of low pressure. And when we think about pressure, I'm going to draw a circle. So when you think about pressure, if you think about stuff inside a compartment, if you have a lot of stuff inside, right, we're thinking about it as pushing on the walls. So like that would be higher pressure than just a little bit, right? It's not exerting as much pressure. The other way we can change pressure is by changing the size of our compartment. So if we suddenly have a contraction, and now we still have that same amount in there, this is going to be higher pressure because the size of that compartment has changed. So just conceptually, that's what we're thinking about when we're talking about pressure. So as we put blood into the right atrium, it's raising the pressure because we're putting more stuff in there. As that pressure rises, nothing's happening to the ventricle yet. So it's staying the same amount of pressure it started at. As the pressure in the atrium is rising, it's now getting higher than the ventricle, so blood starts to flow into the ventricle. So that part is pretty passive. And then once the ventricle contracts, they're changing the size of that compartment, which means that the pressure is rising as that space gets smaller. And this is what pushes those AV valves to close, right? So we think about the blood physically here and the contraction of that muscle as pushing back towards the right atrium as the pressure is rising. So this pushes the valves closed. And as those valves close, this is strong as we keep contracting that pressure. So they might have a tendency to prolapse back into the atrium, right, and open up that way. But we don't want them to do that because blood flow has to be unidirectional, right? We're trying to guide it to the lungs. If it just pulls back in a previous step, we don't get it oxygenated. So here in the right atrium, we have these papillae, these papillary muscles connected to the valves by these little cords, these chordae tendineae. And that just exerts like a tiny little pull on the valve to keep it from going into the right atrium, so that when it's in its closed state, it would just like create a wall, right? So in an open state, the valves would look like that. And in a closed state, we're just physically forming a wall across, right? What we don't ever want to happen is to have them go like that. That's not a state your valves should ever be in. Right. So the papillary muscles are preventing this third state. Does that make sense? So the other type of valves we have are the semilunar valves. So we refer to them based on their locations, the aortic valve or the pulmonary valve. And what they look like, you can see the picture here, but I'm going to draw out the aorta, let's say. Okay. So here's our aorta, curved structure. Our semilunar valves are like little cups in the wall. Okay. And if we were to look down on them from above, they're going to look kind of like this, like little leaflets closing over. You can see how they form this kind of bag-like structure. So what's happening with those semilunar valves is so when we want blood to flow into the aorta, these valves, we have the left ventricle contracting, say, because we're looking at the aorta. So this blood pushes on them, right? And they collapse back against the walls of, in this case, the aorta. So they would just flatten themselves and press against the walls, right? So we have an opening. So we're able to move from this area of high pressure in the ventricle to lower pressure in the aorta here. Now, once this happens, as we move blood into the aorta, that builds the pressure in the aorta, because as you put more stuff somewhere, that raises its pressure. So what we have happening, once we have high pressure here relative to pressure in the left ventricle, the blood wants to flow backwards. So it starts to flow towards those valves, and these cups catch that blood and inflate it. So we go from a situation like this, as the blood flows back, to the closed state. So they actually come in contact with each other as those cups catch the blood. So this is why we don't have blood backflowing into, in this case, the left ventricle. Same thing happening with our pulmonary arteries in the lungs. So the difference here is that we don't have those chordae tendineae. So if you were to physically look at these valves, they look different. The shape is different. So go ahead and draw me some valves inside the heart. Label them and draw some pressures and blood flow. Okay. Got a heart looking something like this with our AV valves. So right AV valve, our tricuspid, left AV valve, mitral valve, their job, unidirectional blood flow, as we end up with high pressure in the ventricle relative to lower pressure in the atrium. We want them to stop blood from backflowing from the ventricle to the atrium. We'll skip that for now. Oh, actually, we're up to the electrical activity. So go ahead and think about this one, I guess. Okay, so when the ventricles contract, are the valves open or closed? And you can just yell it out. During systole, when we're contracting those ventricles, are AV valves going to have to be open or closed? What about the semilunar valves? If we have our ventricles contracting, what about those valves in the arteries? Are they going to be open or closed? Yeah, perfect. Okay, so next time we'll talk about how this gets controlled using electricity.
Kaya, is the Zoom recorded? I didn't get like the first 10 minutes of the lecture.
It's up on YouTube.
All right, sounds good. Thank you.